Citronella essential oil is widely used and applied to the skin, whether as an insect repellent, astringent, or aromatherapy. But do we ever imagine, do this essential oil affect the process of our skin aging? I, Denaya Kumara, Kusnul, Unica, and Dr. Muti Ikawati present our research anti-aging properties of citronella essential oil by targeting PTGS2, CYP19A1, and HMGCR in cellular senescence pathway. Several studies have shown that citronella essential oil has many properties such as astringent, antibacterial, and antioxidant. Antioxidant activity could reduce high level of ROS that can be triggered by the surrounding environment and daily lifestyle such as UV rays, stress, pollution, and diet. High level of ROS could induce the cellular senescence, and the cellular senescence process is one of the causes of skin aging. Inhibition of cellular senescence by citronella essential oils' antioxidant properties could potentially prevent premature skin aging. Therefore, this research was conducted to explore the antioxidant activity of citronella essential oil and to reveal its ability to inhibit cellular senescence that causes premature aging of the skin. The target proteins and antioxidant activity of compounds from literature study were predicted with Swiss Target Prediction website. We retrieved data about proteins that contributed to cellular senescence from GeneCards database. From those two databases, we used interactive and an online tool to intersect the two previous data so we can analyze which target proteins are involved in cellular senescence. The data were further analyzed with NIME and MOE software to predict citronella essential oil constituents activities and interactions with its target proteins. There were six most abundant constituents: citronellal, geraniol, citronellal, nerol, and nerol. The abundance of those constituents were more than 10%, so it is expected to give significant effect in skin aging prevention. All of those constituents targeted oxidoreductase protein, shown by green color on the pie chart, based on chemical structure prediction using Swiss Target website. Moreover, shown by the yellow color, some constituents also targeted cytochrome P450 proteins that also contributed to oxidation reduction process inside the body. These two protein classes, oxidoreductase and cytochrome P450, indicated that the dominant activity of citronella essential oil constituents involves oxidation reduction process. This prediction might be the underlying causes of antioxidant activity of citronella essential oil. Based on GeneCards database, there were thousands of protein coding genes contributed to cellular senescence. Those proteins were intersected with previous target proteins that underlie antioxidant activity of this essential oil. The intersection revealed that there were three proteins that were involved in antioxidant activity and also cellular senescence process. Therefore, citronella essential oil could potentially prevent skin aging with its antioxidant activity by targeting those three proteins that were involved in cellular senescence. Activity prediction with NIME software revealed that 4 out of the 6 constituents, geraniol, citronellal, and nerol, were predicted to inhibit PTGS2, CYP19A1, and HMGCR proteins with high prediction scores. Prediction models for each protein have near 100% overall accuracy. Underlying molecular interaction were further analyzed using molecular docking. Geraniol, citronellal, and nerol bound to PTGS2 with a close binding affinity to that of selective PTGS2 inhibitor rofecoxib. Furthermore, geraniol and citronellal have shorter binding distance with PTGS2 residue, which indicated stable interactions. Meanwhile, citronellal and citronellol interacted with the same CYP19A1 residue as exemestane, a CYP19A1 inhibitor, although the binding affinity was lower than that of the inhibitor. Moreover, citronellol bound to more than one amino acid residue, so it could strengthen the interaction between the constituent and CYP19A1. On the other hand, geraniol and citronellol bound to several residues of HMGCR, which were also the target of simvastatin. Despite having a lower binding score, citronellol bound to HMGCR residues with a lower distance than simvastatin, so that a stable interaction can be formed. Through the aforementioned interactions, the inhibitory activity against PTGS2, CYP19A1, and MGCR proteins that were involved in cellular senescence indicated citronella essential oil potential in preventing skin aging. All of the results revealed that citronella essential oil constituents were predicted to have antioxidant activity by inhibiting CYP19A1 and HMGCR proteins that induce the formation of reactive oxygen species and also inhibiting PTGS2 proteins that could increase the risk of chronic inflammation. Decreasing levels of excess ROS with citronella essential oil could prevent the cellular senescence process so that premature skin aging could be prevented. This research shows that citronella essential oil possibly prevents cellular senescence through its antioxidant properties by targeting PTGS2, CYP19A1, and HMGCR. Hence, it can potentially be developed as an anti-aging agent. Thank you.